I'm going to get started, if that's all right. We're very happy to have Dr. Laura Case visiting us here today, and Laura has been to San Diego before. She did her doctoral work with Dr. Ramachandran in the Department of Psychology, and her dissertation was How the Body Can Feel Wrong, Sensory Processing and Neural Body Representation in Anorexia and Transsexuality. Super interesting topic. That was in 2013, and then Laura went away to Bethesda. Are you in Bethesda there? And she is a postdoctoral fellow at the National Center for Complementary and Integrative Health, which is within the National Institutes of Health, working primarily with Catherine Bushnell. And Laura has expertise in functional magnetic resonance imaging, and her work transcends a lot of different very interesting areas, including sensation and into pain and eating and gender behaviors, gender identity and eating behaviors in particular. So very interesting work. So we're really excited to hear about your presentation today.
Thank you. Can everyone hear me well? Okay, great. So thank you for the invitation. I'm so happy to be here and share my more recent research with you. Okay, coordinating my clicks. All right. So my research on affective touch began with an early interest in mind-body interaction, how who we are fundamentally shapes our perception of the world. In my doctoral research here at UCSD, I studied people with phantom limbs, people with eating disorders, and people who were transgender. All of these individuals perceive some significant discrepancy between their physical body and their mental representation of their body. But this mental representation wasn't just an image in their head. It profoundly altered how they experienced bodily sensation and whether it felt good or bad. People with phantom limbs experienced painful sensations originating from a limb that wasn't present. People with eating disorders felt larger than their actual size and experienced distortions in processing tactile sensation. And people who are transgender often experience touch on certain body parts as feeling incongruent and distressing. I was fascinated by the role of body perception and clinical conditions in perceiving touch sensation. How is it that two people can receive identical sensory input and it can feel completely different, both physically and emotionally? This led me to pursue postdoctoral research with Dr. Catherine Bushnell at the NIH on how touch and pain are processed in the brain and modulated by psychological factors. In particular, I have focused on sensory pathways that lead to a sensation of pleasant touch. And I've also studied how top-down factors in the brain influence touch perception, including mood and expectation and neurochemistry. To perform these studies, I've used interdisciplinary methods, including behavioral testing, brain imaging, non-invasive brain stimulation, and pharmacology. And I've worked with healthy individuals and those with significant sensory abnormalities. Putting these interests and expertise together, my broader vision as an independent investigator is to elucidate the mechanisms by which sensory input and top-down psychological factors impact the perception of touch. I study how these mechanisms differ in various clinical conditions. So for example, studying touch perception in people with chronic pain can help us understand both the mechanisms of chronic pain as well as the mechanisms of manual touch therapies, which patients can benefit from them, and how to improve them, which I think is very important given the current opioid epidemic and the search for non-pharmacologic treatment alternatives. My vision overall is to understand the experience of affective touch and how this interacts with clinical conditions like chronic pain. So today I'll give you a snapshot of my research in several of these areas. I'll start with a study on sensory input, or two studies, one from the skin, one from deeper tissues like the muscle. I'll then give an example of my clinical research and tell you about how chronic pain alters pleasant touch perception. And then for neurochemistry, how endogenous opioids affect touch perception. And I'll conclude with research directions and broader goals for my future research program. All right. So the perception of touch is often broken down into aspects of touch discrimination and touch affect. So discriminative touch is detecting where and when something has touched our body and what this was, which helps us navigate the environment and identify objects. So this includes features like location and texture, So for example, the sharp claw of a crab or the soft skin of a baby, the softness and the sharpness. In contrast, affective touch is whether the touch feels good or bad. So the pain from the crab pinching us, or the pleasant, rewarding feeling potentially from the baby's hand. Pleasant touch is fundamental to human social relationships, to romantic relationships, to parent-child bonding. Children deprived of touch show stunted cognitive and physical development. And even between strangers, touch increases trust. So if a waiter touches you on the shoulder while delivering the check, tips increase by nearly 50%. Touch is central to grooming relationships in non-human primates. And then in our species, in human primates, we have ritualized forms of touch like massage therapy that was a $12 billion industry in 2015, which I think is evidence that pleasant touch is highly motivating and rewarding. We know a lot about how discriminative touch is processed here in primary somatosensory cortex to begin with, but much less about affective touch and in particular pleasant touch. So I'll start with my work on how the brain perceives sensory input as pleasant when it originates from the skin. If we think about why touch feels good, there are two sets of factors. There's the physical and there's the psychological. Psychological factors could include your expectations, your hopes, the person, your personal knowledge, your mood, and so on. So the same touch can feel very different depending on context. So for example, a pat from a caring doctor could feel warm and reassuring, while the same physical touch from an airport screener could feel uncomfortable or alarming, depending on your state of mind. Then there's the physical touch itself. So different somatosensory stimuli activate different populations of sensory afferents. And just as we have pain fibers that code for a harmful stimulus, certain sensory fibers may code for a beneficial touch stimulus. So for example, a number of studies now suggest that a subset of C fibers called C tactile fibers are especially important for pleasant, light touch. And this is what I'm studying currently, these bottom-up sensory contributions to touch affect. All right, so think for a minute about petting a cat. There's a right way to pet a cat and a wrong way to pet a cat, right? So we all know this, but why is it? What is it about slow, gentle stroking on the cat's back in the direction of the fur? Well, the way a cat likes to be pet has a lot to do with C tactile sensory fibers. We have two kinds of afferent sensory fibers in the body. A fibers, which are myelinated, and they conduct their signal very quickly to the brain, and C fibers, which are unmyelinated, they're in orange, and send their signal much more slowly, taking about a second to reach the brain. C fibers were thought to only conduct dull pain and temperature sensations, but they've been found in more recent years to also substitute them to respond to gentle, slow stroking. This has been found in cats, rabbits, rats, monkeys, and it was believed for a while they were absent or present only in a vestigial form in humans, but then they were identified using micronography techniques in 1993, so we have them too. CT fibers are tuned in the periphery to respond to slow, gentle stroking on hairy surfaces of the body, which is essentially everywhere aside from the palms of the hands and the feet. So this is data from one of my studies. It is the same finding as nearly every other study of C tactile fibers, that if you compare the ratings of slow and fast brushing, people find fast brushing much more intense, but slow brushing much more pleasant. A fibers project, change this as well. All right. A fibers project to early somatosensory areas, so first to primary somatosensory cortex, while the C and CT fibers project directly to the S2 and insula area into the cingulate cortex. This has led to a theory that these separate touch pathways, or largely separate A and C, in the periphery are also coded separately in the brain and subserve separate functions. So the A fibers for touch discrimination, that where, what, when, and C fibers for the more affective aspects, the pain and pleasantness. And we have a lot of data about discriminative touch and its processing in S1 and S2 in the insula, but much less on affective touch and in particular pleasant touch. Are these early sensory areas involved, or is it a more frontal and limbic representation? However, a couple recent studies contended that S1 is also critical for perceiving these affective parts of touch, the pleasantness. But these studies had a number of confounds. So if you think about it, it's very hard to change the pleasantness of touch without also changing some other physical features. So some studies will do something like present a sharp stick versus some soft velvet. Of course, the soft velvet is more pleasant, but it's also a different texture, a different intensity, and this is often not controlled for. Other studies will use a cognitive manipulation of touch, so the same touch, but you have a picture of who's administering the touch, somebody you like and somebody you don't like. And this showed an S1 modulation, but there again, some confounds in the task I can go into if anyone's interested in terms of attention and motivation. You're supposed to be looking for a date while you do the task. It's a little complicated. So, it's very hard to disentangle, especially if you're not measuring both. So I took a different route, which is to use non-invasive brain stimulation to modulate the level of activity in one of these brain areas and then see does that affect the touch discrimination or the touch affect. So the question is, does S1 play a causal role in touch pleasantness? And the prediction is that inhibiting S1 will alter aspects of touch discrimination, but not aspects of touch pleasantness. So the technique I used in this study was repetitive transcranial magnetic stimulation, or CMS, which is a brief magnetic pulse that affects the firing of neurons within about 2 centimeters of the scalp. So it's on the order of strength of the inside of an MRI scanner, but very transient and fairly focal. I use repetitive TMS, which is a regular pulse sequence, which has longer lasting effects in a single pulse. So you get a behavioral window of maybe 10 minutes after, say, 20 minutes of TMS. And I used a 1 hertz frequency, which is in a range that has been found to have an inhibitory effect on cortex, making it less likely for neurons to fire. So in this study, TMS to S1, I had healthy adult volunteers come in on 2 separate days. And on each day, they received 20 minutes of inhibitory TMS. On one day, it was aimed over the right hemisphere S1. On the other day, it was aimed over the vertex, which is a point on the scalp that's furthest from neural tissue, and effects on the cortical tissue are rarely to never seen. Before and after each TMS session, I conducted sensory testing. So my first task was essentially a positive control of 2-point discrimination, which has been shown previously to be affected by this paradigm. So this is a task where you are discriminating between 1 and 2 points on the skin. So if you can tell a difference, that's a better performance. And then I had my affective task. So ratings of slow and fast gentle brushing rated on visual analog scales. So this is to look at the changes in intensity perception and changes in pleasantness perception. Okay, so I'll show you the data from the spatial discrimination task first. Here you can see percent correct. This is from before to after the S1 TMS, and this is in the vertex condition. So you can see there's a significant interaction, a significant decrease in the ability to discriminate 2 versus 1 after inhibition of S1. So this validates the paradigm, essentially. Next, we did see a tendency towards a difference in intensity perception. This was a significant change from before to after in the S1 condition, but it was not a significant interaction. So it's kind of weak evidence of a change in intensity perception. And finally, no change in touch pleasantness. So aspects of touch discrimination, spatial discrimination, are all altered, but not touch pleasantness, which fits with most of the literature that affective touch is not in these early somatosensory areas. So next, I asked a similar question about S2. S2, secondary somatosensory cortex, implicated in attention and learning, detection of changes in the sensory domain, and is sometimes implicated in affective processes as well. So I again conducted 2 sessions of repetitive CMS, one targeted over the right S2 and the other targeted over the vertex, this time with a foam spacer. I'm happy to go into details if anyone's interested, but this was sort of a novel variation on a procedure to control for the sensory properties of the stimulation without having any cortical effects. And this is because here we're using a deep TMS coil, an H8 coil. Part of our hope was to reach the insula and look at effects of insula inhibition, but our group and several others at NIH simultaneously testing this insula deep TMS coil found it didn't quite reach the insula. So it's an S2 study. Again, I conducted sensory testing before and after the TMS, but here, since I didn't have a great behavioral assay for inhibition of S2, I walked participants across the hall from the TMS room into the scanner right after to check for changes in sensory response, in the bold response, to validate that I had actually inhibited S2.
Question.
Yes.
So it sounds like what you do is 20 minutes of TMS,
Mm-hmm.
and then 10 minutes of behavior, and
Yes.
then into the MRI.
Yes.
MRI.
Yes.
So that introduces a bit of a confound to what's going on during the behavioral testing might not be reflected in the fMRI.
Yeah, that's true. The primary outcome was the sensory testing, so we prioritized that. And by the time they get to the scanner, that sensory session could override some of those TMS effects. But it was within subjects on 2 different days, so the sensory effects should be the same on either day. All right, so this is showing you the main effects of the brushing after the vertex session, the control session, and then after the S2 inhibition. So here you can see a nice brushing evoked response in secondary somatosensory cortex. Here you can also see a response, but it looks less strong. This was not significant in a whole brain analysis, so I did a constrained ROI analysis of peak brushing evoked response areas that had peak response to brushing in our previous studies. So here I'm looking at right and left S2, S1, and the posterior insula, where our peaks of activation were in a previous study. So here you can see that there is significantly less brushing evoked response after the S2 session compared with the vertex session. This was also the case in left hemisphere S2. Not too surprising given the transcolossal connections and strong bilateral connectivity between those areas. But there was no significant change in S1 or in the insula. So this suggested that we successfully turned down processing in right S2. As for the behavioral results, I had predicted decreased intensity perception based on a prior TMS study showing decreases in pain perception after inhibitory TMS to S2, and that is indeed what I saw. So I saw a significant decrease in intensity perception of gentle brushing after S2 inhibition. Although it was only for fast brushing, we didn't see an effect for slow brushing. Fast brushing is the more intense stimulus, and no changes in pleasantness. So in sum, deactivating S2 or S1, partially deactivating, did not alter the pleasantness of gentle touch, despite altering spatial discrimination or intensity perception. So that leaves us with the question of where in the brain is pleasant affective touch processed? To answer this question, I'd love to do deep brain TMS very focally, but it's just not quite there yet. So I conducted an fMRI study where I correlated brain response with perceptual ratings of intensity and pleasantness to look at which brain areas underlie touch pleasantness when controlling for actual intensity perception. So in this study, this was 26 healthy adults. I administered blocks of fast brushing and slow brushing on the palm and back of the hand and collected ratings on visual analog scales for intensity and pleasantness. All right. So first I looked within subject at correlations between intensity ratings and whole brain activation, and I saw that stronger ratings of intensity significantly predicted activation in S1, bilateral S2, and the posterior insula. So this fits very well with the literature on perception of intensity. Looking at correlations with ratings of pleasantness that were significantly greater than correlations with intensity, the only area I saw was the pregenual anterior cingulate cortex. Now this was really exciting because this is an area that is highly opioidergic. It is seen in a number of studies of placebo analgesia, and it also has been linked to pleasantness perception in the domains, sensory domains of taste, smell, and temperature. All right. So in some, I found that inhibiting S1 or S2 altered aspects of touch discrimination or intensity, but not the pleasantness of touch. And touch intensity correlated in fMRI with activation of S1 and S2, these same areas, but pleasantness only with the pregenual ACC. All right. So I want to tell you next about my research on perception of affective touch from deeper tissues. So as I was doing this research on C-tactile perception, I thought a lot about what other types of touch are pleasant. And one that really stood out to me was deep pressure. So deep pressure is perceived as pleasant and relaxing in a number of contexts. The swaddling of infants, hugs, deep tissue massage, and there's even physical occupational therapies for compression, deep pressure, often for children with anxiety, autism, and ADHD. And lots of kind of new age products. You can buy a sofa that will give you a big hug at the end of the day. So there's a lot of evidence that deep pressure is pleasant and relaxing. Some of these clinical studies of compression vests and weighted blankets. And there's some data on parasympathetic nervous system response, but not really anything known about the mechanism. So you might wonder if you can perceive deep tissue, if you can actually perceive sensation from your deeper tissues. It feels like you can, but is that just an illusion? Is it really a cutaneous-based sensation? Well, there's one study that addresses this pretty well. So Graven Nielsen and colleagues found that after blocking cutaneous sensation with a topical anesthetic as well as a subcutaneous injection of lidocaine, they found that the ratings of intensity for deep pressure were preserved at least about 90% of the way. So this strongly suggests that indeed we can feel sensations from our deeper tissues. And of course, we can feel pain from our muscle and deeper tissues. There's work on that. So I hypothesized that there might be a distinct sensory afferent pathway to convey this affective part of deep tissue pressure feeling good in the same way that there is this cutaneous C-tactile pathway for the pleasantness of light touch. Now, I wanted to start with the bottom-up sensory affective input and not the kind of interpersonal context of massage therapy, which adds a lot of complicating factors. So I wanted to design a way to administer pleasant deep pressure that was not social. So what I came up with, this is a compression sleeve from a lymphedema pump. So it has different chambers. It could just go. It's like a big boot that goes up the leg. And I connected it to a compressed air tank. And this is a custom in-house device I collaborated to design with our machine shop so that I can program the flow of air by voltage and regulate the airflow into the sleeve. So I conducted some pilot studies to kind of identify parameters of this touch that would feel good, basically. So I'm happy to talk about that if anyone's interested. But what we came up with was sort of a oscillating pressure. Is much preferable to a static compression.
Why not use something simple like a blood pressure cuff?
Yeah, good question. Mostly because I can't program it. That's kind of what this is. Yeah. it has like the nice multiple chambers, and it's very similar. Yeah.
This sounds like the squeezers they use for deep venous thrombosis.
It is. It is that. But I cut it off from the box they use because they have like a very set sequence. So I want to be able to set my own pressure and frequency. And all right. So first I wanted to see if this was generated a pleasant affective response. This is my colleague Håkon Olausen, who's performed a lot of the C-tactile research, microneurography recordings, and he enjoys it, which is was so exciting. And our healthy participants agreed. So this is 25 individuals rating adjectives that have been found to describe the range of affective responses to touch. And there you can see that they endorse very similar words. So comfortable, soothing, calming, relaxing, enjoyable, pleasurable are endorsed at very similar rates for pressure in blue as for brushing in green. In contrast to painful heat in orange is just rated as not comfortable. All right. So going back to mechanism, we have evidence that the deep pressure sensation is probably based in muscle or other connective deep tissue afferents. But animal and human studies have found conflicting results as to whether it's large or small diameter fibers that underlie innocuous levels of pressure perception. We actually don't know. So I was fortunate enough to be able to test two neuronopathy patients, very rare neuronopathy patients, who had very unusual consequences from either viral or autoimmune conditions in their early adulthood. They're now older adults. They've been studied across the world in sensory motor labs. Both of these individuals have no proprioception, so they cannot walk without extensive training and visual feedback for one of them, but the other is wheelchair bound. And they have no sense of touch aside from heat and pain, and a little bit of the C-tactile pleasant sensation, which has been researched in them. So I wanted to know, would they be able to perceive innocuous levels of deep pressure? Because if they could, that would suggest that it is reliant on C fibers, which seemed likely given the C-tactile pathway. Okay, so the first question, can these A-beta diaphragm patients detect innocuous levels of deep pressure? Well, the task I used was a two alternate force choice task where I inflated the compression sleeve to about 30 millimeters mercury and then 60 millimeters mercury, just paired, and asked, can they tell which is the stronger pulse? So a difference of 30 millimeters mercury. I had 4 age-matched healthy older adult controls, and all of them were 100% of this task, so it was very easy. In contrast, the 2 A-beta diaphragm patients were at or below chance being 50% on this task. So they were completely unable to discriminate the pressure, and when asked, just said, did anything happen? Has it started yet? Like nothing. So I turned the sleeve up to a difference of 90 millimeters mercury, which is the difference between like a small squeeze and a very strong squeeze, but not painful, and still absolutely nothing. So this strongly suggested that A-beta fibers underlie innocuous levels of innocuous deep pressure sensation. So next, I wanted to know, can they perceive noxious levels of deep pressure, and at what point will they perceive deep pressure? So here, I did switch to a blood pressure cuff because my device is not rated for such high pressures. And I simply manually inflated the blood pressure cuff, asking the controls to report when they felt any pressure sensation and then when they felt any pain. So they felt the pressure sensation right away. Somewhere on the order of 10 millimeters mercury, but they didn't feel pain until above 100 or 200 millimeters mercury. In contrast, the 2 patients couldn't perceive the deep pressure until nearly 200 millimeters mercury, like the level that you'd inflate a blood pressure cuff to, and then they perceived pain a little bit after that, probably at the normal level for noxious perception, but I would need some more data from healthy controls to confirm that. But certainly they're not feeling it until it reaches a level that is normally painful. So probably intact nociceptor, afferent nociception from the deeper tissues, but not non-painful pressure sensation. This is just to show you that the device I have is MRI compatible, and I've been imaging the pressure sensation all year. This is just preliminary data. I have a lot more to collect and analyze. Seeing very nice activation of S2, and I'll be looking, when the study is complete, at representation of deep pressure touch as compared to the gentle brushing cutaneous touch. All right, so to sum this study up, most healthy adults like deep pressure sensation and find it calming. Non-painful pressure sensation appears to depend on A-beta fibers. And next, I want to experimentally confirm this in healthy volunteers. And to do that, I'll be using nerve blocks. So this is one of my K99 studies. I've mostly finished one of them. This is the other one that I'm starting. And here I'll be administering nerve blocks within subject design, where I, with an anesthesiologist, either block C fibers using like an injection of lidocaine, or block A fibers using an ischemic compression block, so applying a tourniquet. Of course, this is like a differential block, so either block will affect one fiber type more strongly and first. And then I can administer sensations over time and look at the loss of sensation correlated with the C fiber or the A fiber function, and test where deep pressure is, when deep pressure is still perceived. So for example, if cool fibers are blocked, but warm sensation persists, then we know the A fibers are blocked, but the C fibers are still online. So I hypothesized that blocking the A fibers will lead to decreased pleasantness and intensity of pressure, since the whole perception of this sensation appears to be A beta. All right. So I'll return now to the question of what underlies the perception of pleasant touch in the brain. So recall that in my prior study, pleasantness ratings were correlated with activation of the pregenual ACC. Now, this is a region that is highly opioidergic, and it's involved in endogenous pain modulation. It also differs in people with chronic pain. So for example, my colleague Markou has found significantly decreased gray matter in this exact region in patients with fibromyalgia. So this led me to wonder about the role or the effect of chronic pain on pleasant touch perception. And I was able to test this in individuals with fibromyalgia, which is a condition involving widespread pain, fatigue, and cognitive difficulties. And since it, never mind, okay. Another feature of fibromyalgia is decreased central mu opioid receptor availability, which has been linked in animal studies to decreased density of opioid receptors, but may also relate to hyperactivation of the endogenous opioid system as a means of relieving the ongoing pain. So I hypothesized that chronic pain would alter the perception of touch pleasantness. This study was a collaboration with Markou, who was studying individuals with fibromyalgia. And in this paradigm, participants rated the intensity and pleasantness of slow and fast brushing. So these are the results I showed you before from the healthy individuals who find that fast brushing is significantly more intense, but slow brushing is significantly more pleasant. In contrast, the patients with fibromyalgia showed no differentiation between slow and fast brushing in terms of its intensity, and no differentiation at all in terms of pleasantness. So they're having a blunted affective response to touch. Not completely diminished affective response, but lacking that C-tactile preference. Okay. So this led me to wonder about the role of endogenous opioids in touch perception, given the differences in opioids in chronic pain and the highly opioidergic nature of the PGACC. Another reason to suspect involvement of opioids in touch pleasantness is that there's actually quite a few studies in monkeys where naloxone or naltrexone has been administered to block endogenous opioids, and the monkeys have sought more grooming and sustained that grooming for longer. So they seem to want grooming more, but we can't really ask them what they're feeling, so I decided to try this in humans. So I hypothesized that endogenous opioids would underlie the pleasantness of touch, and specifically that blocking the opioids would make touch more pleasant. This is the endogenous opioid system, or the mu opioid, one of the prevalent opioid receptors. It's found throughout the central nervous system as well as in the gut, and it can be blocked by a competitive antagonist like naloxone. All right. So this was actually part of the same study I just told you about. So there's the baseline brushing task, and then participants were randomized to receive an IV infusion of either naloxone or saline to look at how blocking the endogenous opioid system affected perception of pleasant touch. So we repeated the same brushing task after the naloxone or saline administration. The first thing that I found was that blocking the endogenous opioid system increased touch pleasantness in healthy volunteers. Now, this was a significant increase within the naloxone condition and marginally significant looking at the interaction. This did not occur in the chronic pain patients, however. What was really interesting was that there was no change in touch intensity for the healthy volunteers, but a very significant interaction in the fibromyalgia patients, where blocking the opioids decreased the intensity of the touch. So this is something that I want to follow up on. I am currently 80–90% through with the study. Unfortunately, I'm still blind to it, so I can't share the results, but I'm hoping that that finding will replicate. So this is a within-subject crossover double-blind study that's better powered to look at these differences, and it's in healthy volunteers who come in for 2 separate sessions. One session they get IV naloxone, the other they get IV saline, and here I have fMRI, so I'll be able to look at the brain areas that may mediate this effect of opioids on touch pleasantness. I also am administering deep pressure and also heat pain during this paradigm, so I can compare responses to C-tactile pleasant touch, to deep pressure pleasant touch, to pain. I think this will be interesting because, well, many reasons, but one being that naloxone, by blocking the endogenous opioid analgesic response, so naloxone tends to increase pain perception. You might think that would decrease pleasantness perception, but the data I showed you and the data from the monkey studies would suggest the opposite. So my theory is that perhaps it's low levels of opioids that make the seeking and rewarding aspects of pleasant touch more pleasant, and when there are high levels of opioids, that effect is absent, as in chronic pain. Okay, so to sum up, chronic pain blunts the C-tactile touch preference, and endogenous opioids alter perception of C-tactile touch and do so differently in chronic pain than in healthy volunteers. All right. So I'll tell you now about my future directions for my broader research plans. In the R00 phase, I plan to study why pleasant touch is different in people with chronic pain and the role of opioids. So essentially running the study I've been running this year in healthy volunteers with naloxone and fMRI to see, is it really the case that that pleasant touch is blunted, is it specific to C-tactile touch, but also looking at deep pressure and pain and whether the effects of naloxone are similar on pain and on pleasant touch are related. I also will be looking at psychological factors that might mediate or modulate this relationship, so things like depression and trauma history that are highly prevalent in individuals with chronic pain, and opioid history as well. In my future research, I also plan to study the impact of touch on body perception and the impact of touch on clinical conditions like chronic pain. For chronic pain, there is there are a couple studies that show an analgesic effect of deep pressure. And we also know that chronic pain patients, some of them seek out massage and find it to be highly pain relieving. But we don't really know in chronic pain patients what that analgesic effect of deep pressure is. Is it the same as in individuals without chronic pain? And does it depend on the type of pain and so on? There's a study that will come out later this year that I'm aware of that replicates my finding of blunted C-tactile pleasant perception in people with chronic pain in another chronic pain population. And in this study, they look both at people with subacute pain and then people with chronic pain. And they find the blunted touch affect in the chronic pain population, but not the subacute population. So there's something interesting happening probably in that transition from subacute to chronic pain. But the study was cross-sectional, not longitudinal. So I would like to look at what the longitudinal relationship is between pleasant touch and pain. And I could see doing this in individuals with HIV-associated neuropathic pain or other chronic pain populations here on campus. And I'm also interested in the role of psychological factors in that process of pain chronification. Then looking at the effects of touch on clinical conditions, I want to ask whether pleasant touch can decrease chronic pain or decrease that transition from subacute to chronic pain. So this is in the vein of studies using visual feedback, so like the mirror box or virtual reality therapies, also cognitive behavioral therapies that intervene with sort of the learned effect of pain, where after having experience of chronic pain, one develops like a learned association and you sort of start just predicting pain from any type of such touch or movement. And so perhaps pleasant touch can change this expectation. So some interventional work potentially. And then finally work with body dysphoria. So there seems to be a privileged effect of C-tactile touch on body perception. So there's a study of the rubber hand illusion, which is an illusion where you see touch to a rubber hand and feel touch on your own hand, but it's hidden, and you start to feel as if the rubber hand belongs to you, and that touch is happening to your body, and the rubber hand is part of your body. This illusion is stronger, has been found to be stronger when the touch is slow C-tactile touch than when it's not optimal C-tactile touch, suggesting like a privileged relationship between affective touch and body perception and body ownership. But this has not been explored for deep pressure sensation, although I know when I was taught how to administer the rubber hand illusion, Rama showed me how to do some deep pressure, and that seemed to help with the illusion. So I think it would be very interesting to look at the effects of deep pressure on body perception. Are they the same as for C-tactile touch? Do they have a different effect on different aspects of body ownership or body image? After characterizing that, potentially I would want to see whether affective touch, C-tactile touch, or this deep compression could alter body dysphoria. So in particular, with the eating disorders group, looking at interventions for individuals with disordered body image. So more broadly, I aim to elucidate how pain and body dysphoria alter touch perception and how affective touch perception alters body perception, and ways to potentially break into negative sort of feedback cycles to decrease pain, decrease body dysphoria, and have other positive affective benefits. I'd also like to characterize the direct affective benefits of affective touch so that we can explore how they differ in individuals with pain and distortions in body perception. And I hope that this will be helpful to individuals with chronic pain and in the search for alternative treatments for people with chronic pain. So with that, I'd like to thank my mentors and collaborators at the NIH and at Linköping University and NCCIH for my Career Transition Award. Thank you.
We have time for some questions.
The microphone seems very formal for this setting. Oh, okay. So I was interested in the fact that it looked like all of this S1 stuff was done for the right side of the brain. You also looked at S1 on the left side of the brain. But the different sides, especially the insula, have different roles in interoception and other things. Did you choose the right side for a reason? Tell us about that.
Yeah, I chose the right hemisphere because it has been found to have more representation of the body, more spatial affective representation of the body. And I just had to make a choice, so I chose right. But it would be interesting to look at
the left hand.
the what?
Right brain, left hand.
Yes, yes. So I administered touch sensation on the left side of the body so that I would conduct my primary analyses on the right hemisphere. But I mean, I looked everywhere, but the main representations would be in the right hemisphere in S1.
Would you expect similar changes with touch on the right hand, given those cross-callosal connections that you mentioned?
Yeah, I would expect similar changes. I would expect that there's still not a representation of touch pleasantness in S1 and S2, but there might be differences in the insular representation, I agree.
Hello, Laura. Thanks. That was a great talk and great work. So a question, you might know there's some studies out there showing that people who practice meditation regularly have a very different perception of pain than, say, people who don't meditate. How's that working, do you think?
Well, my mentor, Catherine Bushnell, has conducted some work looking at that. So she has looked at effects of well, not meditation directly, but hypnosis on pain perception and shown changes in connectivity between I think it was S1 and S2 for the intensity focus and other regions for the pleasantness. In terms of, sorry, meditation on pain, you said? Meditation effects on pain?
Yeah, pain perception usually.
Yeah.
People who meditate. Yes, people who meditate regularly, you can give them a certain stimulus of pain, and their perception of it is less than, let's say, before they had meditated.
Yeah. So there's some work on that, looking at, I'm trying to think of the author. There's a study on Zen meditators that I actually just was looking at, but finding decoupling between, I think it was prefrontal cortices and parts of the insula or anterior insular ACC. So by sort of practicing a detachment from the sensation or from the affective response to the sensation, saw a decoupling of prefrontal and affective brain regions that probably reflect that kind of distance from the affective response. There's also studies of yoga and meditation showing differences in gray matter in the insula in areas that have also been shown to show impairment after many years of chronic pain. So connectivity would be my short answer. Yeah.
This might be broad, Laura, since you brought it up with hypnotherapy and that there was some work already done on it, is what were the findings on the brain when it came to pain when hypnotherapy was applied, in your experience?
Well, the study that I have in mind was a change in functional connectivity between parts of the pain network that are involved in the affective response versus the intensity response. But that was related to specific prompts to like focus on just the intensity of the sensation or focus on the sort of unpleasant response to it. So changes in connectivity between affective brain areas. So there's, it's sort of possible to decrease connectivity between the sort of first registration of the pain and the kind of affective appraisal step. I don't know a lot about that area.
Well, this was fantastic, Laura. Thank you so much for coming to share with us your really interesting work.
Thank you. All right. So I'll turn this off.